biomarker assay that can give a more accurate diagnosis based on gene expression. A biomarker can be defined as a biological molecule that may indicate abnormal bodily functions. And in this case, we're looking at the increased growth and multiplication of skin cells. Several biomarkers have been linked to melanoma, but our study focuses on secreted phosphoprotein 1 or SPP1 because it has shown to be up to 50 times more expressed in malignant nevi.
We did this by using a nucleic acid assay called loop-mediated isothermal amplification, or LAMP. First, it is loop-mediated because it uses 4 to 6 primers to bind to the target sequence and ultimately form a self-replicating dumbbell structure. One of the inner primers invades with the help of BST, which is a DNA polymerase, and binds to the complementary sequence on the target strand, after which BST will synthesize complementary base pairs. This newly formed strand is then displaced by an outer primer that anneals to an upstream region in the target sequence. As it is displaced, the end of the first strand forms a self-hybridizing loop structure due to reverse complementary sequences in the primer. This annealing and displacement cycle repeats on the opposite end of the newly formed strand with backward, inner, and outer primers. And the resulting product is a short dumbbell structure that forms a seed for exponential LAMP amplification by containing multiple complementary binding sites. 2 additional loop primers are able to bind in the areas in between the inner and outer primers for increased sites of DNA synthesis. And with the following, you'll be able to see a short animation of how LAMP works. So onto our experimental setup, we combined our reagents of our target sequence or negative control, our DNA polymerase, our primers, which are created in-house, our nucleotides, and last but not least, our pH indicator, in which case we use phenol red. And all of this is combined into one reaction tube, which is submerged into an isothermal water bath. And on the bottom and on the top, you'll see 2 results of either amplification or no amplification. To the top right, you'll see that the tubes changed color from magenta to yellow because of proton release due to polymerase activity, and that leads to a pH decrease. And since the pH indicator is sensitive to this change, it'll change color. And on the bottom, you'll see that the solution stays at the magenta color because there is no annealing of the primer to the target sequence. And that means no amplification and no detection of the biomarker. And for our results in conclusion, at least 3 LAMP trials were run for 2 primer sets, and we have here primer sets 1 and 5 with and without loop primers. We've shown data here from primer set 1 with loops. 4 samples were run per trial as shown by the numbers 1, 2, 3, and 4 on the table to represent each of the 4 tubes. And a positive test result was considered when at least 2 samples amplified. The sensitivity and specificity for primer set 1 was 100% and 0% without loops, and 100% and 66.7% with loops, respectively. That means that only primer set 1 with loops shown here produced significant results using a p-value of less than 0.05. There is about a 15-minute time difference in between the positive and negative controls. Some potential issues that could have caused false positives or earlier than expected amplification times in our negative controls include primer dimerization and ambient skin RNA.
So now let's talk a little bit about our future work with this project. So we're still continuously looking for new biomarker targets. However, right now we're specifically narrowing down to this epigenetic phenomenon called DNA methylation, where a methyl group is covalently added to a cytosine base and this thing called a CpG dinucleotide, so just cytosine and guanine, in this region of a gene that's really important for gene expression called the promoter. So if you have a highly methylated promoter, meaning a lot of the cytosines in the CpG dinucleotides have methyl groups on them, then the transcription machinery won't be able to come in and create mRNA, and that's not translated in protein, so the gene isn't expressed. However, if you have an unmethylated promoter, then the transcriptional machinery can come in and make mRNA that's translated into protein, and the gene is expressed. So just remember, methylated promoter, decreased expression. Unmethylated promoter, increased expression. So how do you even detect a DNA methylation? We use this thing called Bisulfite Conversion. So say you have an unmethylated sequence. Here, we're specifically looking at a CpG dinucleotide. If you add bisulfite, that changes the unmethylated cytosine to uracil, which is the counterpart to thymine. However, if you have a methylated sequence and you add bisulfite, the sequence doesn't change. So how you can use this in a diagnostic setting is designing your primers for either PCR or LAMP to cover as many CpG dinucleotides as possible, especially towards the 3' end. So if you have an unmethylated sequence and you add bisulfite, it changes cytosine to uracil, and the 3' complementarity of the primer does not match anymore because guanine doesn't bind to uracil. So the primer won't be able to anneal, extend, and amplify DNA. But if you have a methylated sequence and you add bisulfite, the primer complementarity is conserved because the sequence didn't change. So that GC can bind the CG and it'll extend, amplify DNA. That can be detected through qPCR with fluorescence, or for LAMP, as we discussed, it could be a pH indicator. So it really is as simple a diagnostic flow through as treating your sample with bisulfite, putting it in a tube, and looking for a color change. So how are we even looking for our biomarkers for DNA methylation? We're using online data from these things called methylation microarrays. So the research scientist does this by treating their sample with bisulfite, as we discussed a little bit earlier, and they put it onto a microchip that contains a bunch of DNA probes, which are just single strands of DNA that complement different regions of your genome that end just before that CpG dinucleotide that you're looking for the methylation status of. So how they actually detect methylation, though, is through this thing called one base pair extension. So they take the probe and they see what the next base incorporated into the probe is going to actually be. So if it's guanine, that means the CpG dinucleotide is methylated because cytosine didn't change to uracil. But if it's unmethylated, then the cytosine will change to uracil upon addition of the bisulfite, and then the next base will be added will be adenine. So it's really as simple as tagging guanine with a green fluorescent tag, and then adenine with a red fluorescent tag, and then looking for how the probe fluoresces under a camera. So what are we even doing with all of this data? So we're taking it offline from this huge database run by the NIH called the Gene Expression Omnibus, also called the GEO. Then we're processing it ourselves. We're downloading the raw data specifically, so then we can clean it ourselves. And then we're running this statistical analysis called principal component analysis, or multidimensional scaling, also called PCA or MDS, where it basically plots the variation in gene methylation status between different samples. So we predict it'll look something like this, where melanoma melanoma samples will cluster together on the graph because they're similar in methylation status, and then mole samples will cluster together as well. And then we'll find the biggest sources of variation between these two clusters of samples to determine what our biomarkers will be, and then we'll use some kind of flow chart process like a decision tree to determine the most accurate combination of biomarkers and in which order would be best for a diagnostic panel of biomarkers regarding methylation. So some current issues that we're working on right now include data normalization and file labeling. So the data that we've directly imported into the statistical program we're using, specifically R, is not normalizing very well. So what we're going to try to do is directly download it from the GEO itself and then import the raw data and clean it ourselves, because normalized data should look something more like this. But we're currently facing some issues with file labeling. So when the researchers uploaded their data to the GEO, they didn't necessarily upload it with the correct formatting for R in mind, because that's not always the software that they use. So we're going to have to go in and rename about 300 different files to get it to import into R so that we can run our statistical analyses. But after all that, we hope to have our results and a panel of biomarkers that we can use that we will then test on samples with bisulfite conversion. And here are some of our acknowledgments, and here are some of our references. And if y'all have any questions, we would love to answer them.
So if the judges have any questions, we're going to take those first. And then after that, we can move to the audience.
Yeah, this is Lee Greathouse. For your LAMP amplification, did you decide a priori on a specific polymerase to use for that analysis, or did you just go with what was usually used, or did you try different ones?
We just used BST polymerase just because our lab just, that's what we usually have on hand. Did that answer your question? I'm so sorry.
Yes, it did. I was just trying to get at if that was a variable in your experimental setup to test different polymerases.
I got a question for you guys. So my questions are usually focused a little bit more on a little abstract. How do you guys plan to take the lessons that you've learned as like undergrad researchers to the next level of grad school or med school? Like, what has this taught you skill-wise, work ethic-wise, to move to that next step, to be confident in that next step?
Do you want to go for it, Julia?
Yeah, sure. I think for me, definitely it's persistence and also not just not giving up, because for sure for a lot of our wet lab work, that it was really hard to control contamination, especially ambient skin RNA, and I think just not being afraid to start over, and just redo everything and make sure everything is sterile, make sure that we are either doing aseptic technique correctly or running it under a hood. And really, I think just being able to also enjoy the process as well. And although I am considering going into a health profession school, but I think that that is definitely something that I learned and will take into consideration, because I think that for sure I will want to continue to do research later on.
Yeah, and just to add on to that, you're only seeing 1 of 5 primers to have results. All the other 4 didn't produce statistically significant results, and it's taken a better part of a year to figure out why this is happening. So it really has been a super frustrating process, but we've learned to kind of endure past, as Julia said, the frustration of just not working, and we've learned how to adapt to like these kinds of issues and refine our targets so that we can kind of control for that kind of error, which is kind of what we're doing with DNA methylation.
Okay, and then we can take a couple questions from the audience. I'm going to kind of keep it short because we started a bit late, but if anyone has any questions, feel free to raise your hand or type it in the chat.
I'd like to ask a question as well. Why did y'all do, is there a particular reason why you did LAMP amplification and not PCR? Is that kind of with long-term in mind for like what the clinical approach to this would be? Or is there a reason why going into this, your primers were going to work better with LAMP and that approach? Just going into the experiment, why LAMP as opposed to?
Yeah, also something kind of related to this question that might be in addition to a question that was earlier asked for BST. BST is kind of a polymerase that is specifically used also in LAMP because it has strand displacing ability. So for LAMP, we chose to go with LAMP because it is very cost effective. It is a lot cheaper to run LAMP, especially for our future goal of maybe administering this in point of care areas as a point of care diagnostic. And it also doesn't require really a lot of special reagents or a lot of sophisticated equipment. And another thing is that it takes place at an isothermal condition. So for our water bath, we keep it at around 63 to 65 degrees Celsius. Whereas, for example, if you're going to run even like qPCR, it'll be different cycles at different temperatures. And this also has good efficiency. Another thing is that you don't need to denature the double-stranded DNA, and that it also has a high level of specificity at the same time because it does use 4 primers to recognize 6 distinct regions on the target gene. And I think these are kind of the main reasons. And I think also, especially when considering future applications, you don't really need highly trained personnel to run the reaction. It could be something that just the general public is able to do either inside a clinic or maybe even inside their own homes.
Thanks.
All right, so that is about 15 minutes for their presentation. So we're going to move on to the next one. Our next presentation is by Annabelle and Mackenzie, also from UT Austin. So please share your screen and start whenever you're ready.
I'll go ahead and share my screen. Hi, my name is Mackenzie Noel.
And my name is Annabelle Vincent. We're undergrad researchers at the University of Texas at Austin in the Virtual Cures stream in the College of Natural Sciences.
Today we're presenting our research on Mycobacterium tuberculosis. 139 years ago from this past Wednesday, Dr. Robert Koch announced his discovery of Mycobacterium tuberculosis, the bacterium causing TB. This little bacterium had big implications. In 2016, the World Health Organization, or WHO, estimated that 28,500 people contracted and 4,700 people died from TB every day. About a quarter of the world's population is infected with latent TB, with 10.4 million contracting active TB every year. As seen in the image on the right, severe TB outbreaks are clustered in India, Indonesia, the Philippines, China, etc. In regards to the disease itself, TB targets lungs, but can also affect the kidneys, the spine, and the brain. The disease spreads when contagious individuals sneeze or cough, releasing particles into the air that another person can breathe in. While there is a treatment, a cocktail of antibacterials, many strains of Mycobacterium tuberculosis have become resistant to them. For instance, multi-drug-resistant TB, or MDR-TB, are not killed by the best TB antibacterials, isoniazid and rifampin. As seen in the image on the left, the number of MDR cases has not stopped increasing since 2015. Additionally, strains of extensively drug-resistant TB, or XDR-TB, have also emerged, which are resistant to those first-line antibacterials, as well as the second line of defense antibacterials. Consequently, finding new drugs that kill resistant and sensitive TB alike is critical, and this is what inspired our research.
Now that we've talked about the disease, let's talk more about our organism and the target. So our organism is Mycobacterium tuberculosis, a species of pathogenic bacteria that, as mentioned earlier, was first discovered in 1882 by Dr. Robert Koch. Mycobacterium tuberculosis, pictured in the image above and stained orange, has an unusual waxy coating on its cell surface and a distinct rod shape. Our target is acetate kinase, a protein essential to the bacterium. This enzyme is involved in catalyzing a reaction which uses ATP to synthesize propanol phosphate, which can then be used to facilitate the production of acetyl-CoA. This can be seen in the reaction mechanism in Figure 2. This shorty chain fatty acid can play a major role in the carbon cycle and can be utilized as a source of carbon and energy by the bacteria. We chose this target because of how essential it is to the pathogen. Additionally, while running the amino acid sequence through a protein blast, we found no significant similarity to humans, which makes it a great target to investigate.
In order to study MTAC, or the acetate kinase enzyme of Mycobacterium tuberculosis, we first needed a model. However, there was no X-ray crystallography model that we could study, so we had to create a homology model. First, we took the amino acid sequence of our target enzyme and ran it through BLAST-P. BLAST-P compared our target enzyme's amino acid sequence to other bacterial acetate kinase sequence. The one with the greatest similarity that we found was Mycobacterium marinum, whose acetate kinase was 79% similar to ours. Then we created a homology model. However, Mycobacterium marinum didn't have a ligand docked in its active site. So we had to find another bacterial acetate kinase with the same EC number in order to define the active site of our homology model. Once we did this, we ran it through Molprobity to analyze the structure further.
Molprobity is a program that tests the accuracy and reliability of macromolecule structures, diagnosing potential sources of error in the model. It does an atom-level analysis as well as an in-depth structural analysis to see if the protein model is accurate. Because we created a homology model from a similar organism, Mycobacterium marinum, we wanted to ensure it would be valid to go through our versional screening and docking process. So when running this model, we flipped histone residue 116 and added hydrogens to our structure. As we see in the summary table, we have a green clash score which shows steric interference. Our model scores very highly in the 99th percentile. The red flags in this model are poor rotamers and angles. This means that this is not the ideal homology model and may affect our docking results. But we have to go ahead, as Mackenzie said, there is no X-ray crystallography structure of our original target. but we do take this into account in our discussion later. The next step for our research was to decide on the control ligands we would run to validate the homology model further. So we decided on our control ligands using the Brenda database, picking 10 known inhibitors of the target as positive controls, and through the Zinc-12 search tool, 5 negative controls of similar physiochemical properties.
Here are two tables with Annabelle's and my controls. We ran our controls in both GOLD and ICM, which are programs that can score the strength of ligand and binding interactions to a target protein. Since our active site was not well defined in ICM, I'll focus our analysis on GOLD, which should be more accurate. When comparing my GOLD scores to Annabelle's GOLD scores for the controls, we noticed that our positive controls scored significantly lower than we expected, despite the fact that they are known to naturally dock to acetate kinase. For instance, our best scoring ligand, citrate, scored 45 points, and this is really low for something that is known to dock to the target enzyme. Interestingly, acetate, the ligand that naturally docks in MTAK's active site, was one of the worst scoring ligands in GOLD. In Annabelle's run, acetate scored the 4th lowest, and mine, it scored the 2nd lowest. We expected that acetate would be one of the best scoring ligands because it is the original ligand that docks in MTAK's active site. But we believe that its poor score could be a result of its small size. Acetate is a really small molecule, and so this limits the amount of van der Waals interactions and the number of polar atoms that could participate in polar contacts with the target enzyme. This might explain its low score.
Over a couple of weeks, we ran these controls as well as libraries of over 200,000 small molecules available to us, docking to our homology model using virtual screening programs GOLD and ICM. These programs return scores of binding interaction, a higher score relating to a stronger binding between the ligand and the enzyme. This means the molecule would be a good inhibitor of the enzyme and therefore a strong candidate as a potential drug treatment. We analyze our scores looking at averages, interquartile ranges, and ranges of scores across libraries. So here we have our GOLD scores for all the libraries screened. We can see that our highest scoring ligand scored significantly higher than the 1,000 small molecules we screened. So you can see in the low-pack library, our highest scoring ligand is formoterol fumarate dihydrate, and we will look at this a little closer in our next slide. I'll give y'all a second to look at our figure before moving on.
In this image, we can see our top scoring ligand, and as Annabelle said, it's formoterol fumarate dihydrate. It's a little difficult to see with this image how exactly this ligand fits into the active site, so let's take a quick detour. Here we have a molecular visualization system, PyMol, where we can look at our target enzyme and how ligands interact with it. Again, acetate was the ligand that naturally docks in MTAK's active site. Its polar contacts to the enzyme are shown in the black dashes. GOLD scored acetate as one of the worst ligands overall in terms of its binding strength to acetate kinase. Now, we'll take out acetate and put in our best scoring ligand, formoterol fumarate dihydrate. We can see how well the molecule fits into the grooves of the active site, creating stronger binding interactions. Since formoterol fumarate scored 70 points higher than acetate, we believe that it would outcompete acetate for binding to the target enzyme if formoterol fumarate is present in high enough concentrations. Thus, it could be a strong inhibitor and potential drug treatment for TB.
Now let's take a look at our next steps and discuss our research project a little further. Firstly, we could not find an X-ray structure of our original target, so we created a homology model using Mycobacterium marinum, highly similar organism, as Mackenzie said before. This model has slight differences in its structure compared to the original target and does not score as highly in mole probability as mentioned earlier. Secondly, when running the ICM screening, the program found 6 potential active sites in the target structure. We did not specify which binding pocket the program should run the ligands in, and this is another potential source of error.
Additionally, we hope to run MTAK assays with formoterol fumarate and other ligands that scored well to determine if these could be used as an inhibitor. When researching formoterol fumarate, again, our highest scoring ligand, we discovered that it triggers the increased expression of cyclic AMP, which loosen the bronchial smooth muscles of asthma and COPD patients, allowing them to breathe easier. TB infects the lungs and has ATP as a substrate. We recognize there's a significant difference between the bronchial and tuberculosis targets. One is targeting human cells, one is targeting bacterial cells. So they are very different. However, we thought that the metabolic and positional similarities merited further wet lab research and were an interesting finding of our research. Here are some of the references we used in our research.
Utilizing powerful software, our research found a strong TB treatment candidate that can be further investigated through wet lab assays.
We'd like to thank everyone who helped us in this project. We could not have done this project without all of these individuals, and we're very grateful. The slogan of World TB Day, which again was on March 24th, so this past Wednesday, is "It's time to end TB." That's what our research hopes to contribute to. Thank you so much for listening to our research.
All right, now if the judges have any questions, we have about 4 minutes.
Sure, that was a really nice presentation. I'm not a chemist or biochemist, so it was a nice intro for me into this type of structural imaging. But I did have a question about your wet lab assays that you're planning on in the future. Can you go into a little more detail about how you're going to do this at the bench and these assays that you're going to follow up with?
Annabelle, do you want to answer, or do you want me to?
I can go ahead and jump in. So our wet lab assays are to test enzymatic activity. We want to see if our inhibitors do what they say. It is on paper that they inhibit the activity of our target enzyme. So in wet lab, we would order these ligands that we tested, and we talked about our top scoring ligands, but we also had other ligands that also scored highly, but not as high as our top scoring. So we would get those chemicals or small molecules and run assays with our target enzyme in wet lab to verify that it actually inhibits activity.
To add to that, this research that we're presenting on was just virtual, and so while we believe that this is probable that it could be a good inhibitor, wet lab will help us verify or realize that it's actually not a great inhibitor, and then we'll start over or potentially find other compounds that could act as good inhibitors of this enzyme acetate kinase.
All right, I got my question for you guys. What initially drew you to wanting to work on tuberculosis, and how do you plan on using the skills you've learned in that next step in either grad school or med school?
Oh, sorry, go ahead, Annabelle.
I was going to say, we could split up this question pretty well because originally, because we did this through a class that is held by UT, I hopped onto Mackenzie's target. So if you want to take the first part, I'll take the second part of the question.
Yeah, so I was researching different targets to try to find one that hadn't potentially been as researched as much as others. And I found TB really interesting due to the fact that there is this difference in latent TB versus active TB that we mentioned. Latent TB is where the bacteria is inside of us, but it's present in such small concentrations, and its population size is so small that our body handles it. And what I found interesting was, before this, I didn't know that you could be infected with it, but not really show symptoms or not have the consequences of it. And the fact that a quarter of the world's population, according to the WHO, I believe, is estimated to have that, I found really interesting. And then I have been interested in antibacterial resistance for a bit now. And so learning about the severe or the extensively drug resistant and multi-drug resistant aspects of or strains of Mycobacterium tuberculosis was another reason why I found it interesting.
And the second part of the question was about the skills we learned and how it would translate to graduate school or further research. I think in this lab, we learned so many skills. We were introduced to programs we've never heard of, like Golden ICM, and running those screenings, how to run those screenings, how to start from nothing, like finding a target, all the way to actually getting results and then presenting those results. We presented at the end of the semester to our class, and then Kenzie and I have gone on to present at CTURC, another conference, to this conference, and more in the future we're planning to. So honestly, it's been a whole year of like all the skills that would apply to grad school and further research. I had another point, but I lost it. But that covers it.
I think for me, one of the things, besides learning all these new programs, one of the things that I found most difficult, but also really interesting, was the process of once we had all our data, when we ran it through Gold, we had to go through this process of analyzing our data, as well as figuring out the best way to show our data. And that was really difficult because, as Annabelle said, we analyzed about 200 to 250,000 compounds through these software. So concatenating this data, figuring out how to represent this data was challenging, but also a really cool process at the end to have finished.
You mentioned that you did some blast searches for acetate kinase to make sure that you wouldn't be targeting human cells. Did you notice that that your target acetate kinase was in other common bacteria? Basically, could you be killing off-target bacteria with any of these drugs that you're screening?
I like, in terms of bacteria in our microbiome that could potentially be like a side effect. Okay.
Yes.
I'm not sure specifically on that, but I do know that we saw Mycobacterium marinum had this. We saw E. coli had this. That's really interesting. I'm not sure which bacteria are in our microbiome, so that would be something we should look into.
And then I forgot the name of the candidate that you came up with, but it seemed much larger than acetate. How do you know the Mycobacterium will take it up? Because acetate kinase, it seems like, is acting in the interior of the bacteria. So do you know, will it be too large for it to enter through any of the pores that it has? Or have you looked at like pore size or that kind of? I'm not sure like how far down the pipeline you're looking for that, but just, is that something that y'all have looked into?
Annabelle, do you want to answer, or do you want me to?
Hi. That's a great question and a great thing to look into. I think it's significantly larger in size, and looking back at the physiochemical properties we recorded for all our ligands, I think did we say that it did not follow Lipinski's rule of 5, therefore would not be a great like orally ingested treatment? But Mackenzie was doing further reading into it, and would you like to talk about the aerosol bit, or
Yeah. Also, for those of you that might not be familiar with Lipinski's is this rule of thumb that some researchers use to see, it's like certain criteria, and we see based on these criteria, is our potential compound good to be taken orally, like Annabelle said. And as she said, since ours was very big, it did violate this. One thing that I found was formoterol fumarate, which is this high-scoring ligand, has been used in asthma and COPD patients in combination with other drugs. But how it's taken there, I believe Symbicort is the medicine that combines formoterol fumarate with, it's called busonoside, which is a corticosteroid. And so how they take that medicine is they take it through an inhaler. So it's like a capsule. I'm not really sure how exactly it works, but it's a capsule that you place in this inhaler and then you breathe it in. Again, I'm not sure if that could work in this instance, since that is used to treat the bronchial smooth muscles, and we were investigating it as a drug to kill bacteria or inhibit their growth. But that was one way that it has been used that could go around the size issue.
All right, so we're going to move on now because we are a little bit behind track. But thank you for that presentation. It's very good. And next up, we have Arvind, his first presentation. He is from our own Baylor University. So feel free to share your screen and start as soon as you're ready.
Awesome, thank you. Okay, hey guys, my name is Arvind. I am a sophomore biology major here at Baylor. And today I'll be giving a talk entitled Epithelial to Mesenchymal Transition Proceeds with Dynamic Alterations in CTCF Binding Motifs. So epithelial to mesenchymal transition is basically the process by which epithelial cells, which are the tight-packed cells that line all of the lumens of your body, so all the tubes and blood vessels, basically undergo gene expression and transcriptional changes to become more, to adopt this kind of mesenchymal phenotype. And so mesenchymal cells are generally going to be a lot more loosely packed, freely floating. And this is what allows breast cancer cells to go from primary tumor and spread off via the blood vessels to distant sites. And so metastasis is what contributes to a large portion of cancer-related deaths. And so it's of high clinical importance in order to understand the epigenetic mechanisms by which cells undergo this transition. And so in order to undergo this transition, the cells basically have epigenetic reprogramming by which the actual architecture of the genome changes, and various transcription factors that normally wouldn't be present in epithelial cells or bound to the DNA in epithelial cells then become activated and change the expression of various cell adhesion and ECM proteins. And so my project specifically focuses on the role of CTCF, which is CCC-TC binding factor, which is a protein that basically serves as a molecular anchor. And so CTCF binds to its motif on the DNA, and that's what confers specificity to the chromatin looping conducted by cohesin. So cohesin is this kind of circular protein that anchors onto the DNA wherever CTCF is. And this mediates the large-scale kind of chromatin architecture that's present in cells. And so depending on where CTCF is located on the genome, you're going to have the formation of these large DNA loops. And CTCF can basically bring together promoters and enhancers and form superenhancer complexes that then modulate the expression of nearby genes. And so understanding how CTCF does this is very important to understanding exactly what epigenetic changes are going on to convert a cell from the epithelial phenotype to a mesenchymal phenotype. And so in order to better understand this phenomenon, we conducted Ataxic during various time points of the EMT-MET spectrum. So using TGF-beta, which is a molecule that stimulates a lot of the downstream targets necessary to convert a cell from the epithelial phenotype to the mesenchymal phenotype, we then conducted Ataxic at various time points during this transition, and then we're able to analyze the DNA, the chromatin occupancy during this time. And so looking at how Ataxic functions, basically you have regions of the chromatin that are closed and open. And this TN5 transposase basically comes in and inserts these tag sequences at everywhere where the DNA is open. And you can amplify and read these sequences to basically see where you're having regions of open chromatin. And so the various time points at which we did this is first we had MCF10A cells, which are a mammary breast cancer cell line. And then we had one set that was completely untreated with TGF-beta. and that served as our control. And then we treated the cells with TGF-beta for different amounts of time. And so here we have early EMT, which is 2 days of TGF-beta induction. We have partial EMT, which is 4 days of TGF-beta exposure, full EMT, which is 10 days, transient MET, which is basically the reverse process in which you treat the cells with TGF-beta for 4 days and then withdraw for 4 days to basically convert them from epithelial to mesenchymal. And then when you withdraw, the cells kind of convert back from mesenchymal to epithelial. And we're basically getting a snapshot of that time point. Then we have partial MET, which is a 10-day treatment and then a 4-day withdrawal, and then 2 sets of full MET, which is a 10-day and a 4-day treatment and a 10-day withdrawal. And so from this data, we were able to obtain these Ataxic peaks. And so these peaks are basically going to be regions where you see a lot of copies of those amplified or TN5 transposase cut elements. And so basically what we did here was we took the MCF10A untreated cells, took those Ataxic peaks, and then we took the peaks that we found in early EMT, and we subtracted the early EMT peaks from the control peaks to basically get a peak set that is going to be unique to early EMT. So these are going to be peaks that we see in early EMT, but not in the untreated MCF10A cells. So the rationale there being that the peaks that we see in this peak set should be peaks that are induced by TGF-beta treatment and are involved in EMT induction. And so this is just a screenshot of some of these peaks from IGV, the Interactive Genome Viewer, but all of this was kind of done in Python and R. And so once we got this peak set, we conducted Giggle analysis. And so Giggle is basically a genomic search engine. And so it contains a database of about 47,000 different genome interval files, which are basically these files generated through various different DNA and chromatin accessibility assays. So for example, RNA-seq, ChIP-seq, ATAC-seq, DNA SWAN assays. And so basically, we're able to compare the occupancy of the accessibility of our genome interval file to various different genome interval files that are present on the Giggle database. And so comparing various different time points of EMT to all the different files present on the Giggle database, we can see here in early EMT that we see a couple of top hits for transcription factors involved in hormonal therapy resistance, specifically STAT3, FOSL1, and FOS, June also. And so as we kind of move throughout the EMT, MET spectrum, we see that the transcription factors that are present are dynamically changing. And so, this is kind of just further evidence that we're seeing that the DNA binding factor patterns are changing throughout EMT, MET, and that's what's kind of leading to the phenotypic change that we're seeing. And so a particularly interesting result that we're seeing is that SMAD2/3 occupancy decreases as EMT progresses. And so SMAD2 and 3 are basically direct downstream targets of the TGF-beta signaling pathway. And so in early EMT, which is that 2-day treatment of TGF-beta, we're seeing a high similarity to genome interval files for SMAD2/3 binding. And as EMT progresses, we see that the similarity score goes down. And so what this basically tells us is that at the beginning of EMT induction, we're seeing a transcriptional profile that's largely influenced by that initial TGF-beta signaling, hence the higher SMAD2/3 score. However, as EMT progresses, we're seeing that the initial TGF-beta kind of DNA binding pattern is is kind of demolished by a more complex transcription factor binding network. And so after we kind of looked at this data, we became interested in how CTCF superenhancer interactions change throughout the EMT-MET spectrum. And so currently we're doing some work looking at CTCF and superenhancer interactions throughout the EMT-MET spectrum. So basically what I've done is taken the complete set of enhancers present in MCF10A cells. And from this data set, I've selected all of those containing CTCF binding motifs. Next, I've analyzed CTCF ChIP-seq and ATAC-seq data such that I'm picking out all of the CTCF binding motif containing enhancers that then also have validated ChIP-seq and ATAC-seq signals. And from this highly selected data set, now I can take a look at those enhancers and basically see if there are any SNP variants present at these CTCF enhancer binding sites to see whether there's any kind of like clinical relevance for the CTCF binding at those specific sites. And so when we've kind of cataloged this library of SNPs at CTCF binding motifs, we're planning on doing some base change and loss of function experiments to basically determine whether, to validate whether that single SNP that we're seeing at these CTCF binding sites are contributing to EMT-MET progression. And so we're going to be doing this base change experiment, looking at whether CTCF occupancy changes at that binding site, and then analyzing downstream genes to see whether that influences gene expression, and then also doing a variety of wet lab assays such as scratch assays and cell proliferation assays to basically see whether CTCF occupancy at those particular sites contribute to increased or decreased metastatic ability. And so with that, I'd like to thank you for your attention and open the floor for any questions.
All right, if our judges have any questions, feel free.
So yeah, that was a nice presentation. I apologize for the background noise. I just actually kind of basic questions is, why did you choose TGF-beta as your treatment, and why did you choose MCF10A cells? Because there's many different varieties of breast cancer cells.
Sure, yeah. So we chose TGF-beta as our EMT-MET inducing model just because in the field it's a commonly used EMT inducer. It's also one of the ones that kind of are a little bit less specific, meaning it's not something that specifically targets, just like snail or slug or twist or some of the other, just specifically one of those transcription factors is kind of just, you're seeing a full scale shift towards the mesenchymal transcription factors. And so that was the reason for choosing TGF-beta. And then we chose MCF10A cells because MCF10A cells are kind of a nice kind of happy medium in terms of the metastatic ability. And so they're a mildly kind of metastatic cell line. And so they're kind of the ideal candidates that you'd want to induce EMT in, so that you can kind of see that nice stepwise progression from something that's primarily epithelial and going to be exhibit the characteristics of a primary tumor into something that's more mesenchymal and metastatic. So, yeah.
So what drew you to working in the Tb lab? And I know you said you were a sophomore, so it could change in a couple of years, but is the plan more med school or grad school? And do you want to focus on, in either med school or grad school, do you want to continue to focus on cancer?
Sure. So what drove me to join the Tb lab? I think, so I'm really interested in like stem cell biology as well. And so, although there's not a whole lot of stem cell biology labs here at Baylor, I think the TB lab is a nice place to be because even though the model that we're working with is, breast cancer metastasis, a lot of the kind of things that we study is, like EMT is largely just like a plastic process. And so it's kind of something that underlies a lot of the stem cell biology as well. So I felt that understanding the epigenetic changes in order to basically convert a cell from epithelial to mesenchymal cells is something that will carry over when I'm, studying stem cells, because it's going to involve the same kind of epigenetic changes, the same kind of plasticity that we're seeing in these cancer cells. And so in terms of my future career goals, I'd like to pursue an MD PhD and then conduct research, PhD in stem cell biology, hopefully. So, yeah.
Sweet.
So actually kind of building off of that on stem cells and on Dr. Greathouse's question, how would you take this this kind of experimental model and how do you take this setup to another cancer or to learning more about stem cells? What are going to be the limitations of kind of trying to identify some of these transcription factors and stuff like that if you were doing it on, let's say, a different kind of cancer? If you kind of had a different project, but you wanted to take the same approach, what's going to be specific to your setup now, and what are some of the things that you think you can take to answer another question? It's sure. Like if somebody just said, look at this other kind of cancer, do the same thing. Can you do EMT analysis in that?
Sure. Yeah, that's a great question. So I think moving to different cancers wouldn't really be much of a challenge, just because, in a lot of publications now, in a lot of EMT, MET papers, we're seeing that the journals are asking now for validation in different cell lines and in different types of cancers, just to show that, whatever phenomenon they're describing in breast cancer cells isn't just limited to breast cancer EMT. It's also something that can be seen in EMT of other cancers. And so I don't think there would be much trouble in moving this kind of overall experimental setup to a different cancer. Although there probably would be differences, I'm assuming, in the kind of things that you'd need to induce EMT. Maybe TGF-beta isn't the best option. Maybe you don't want to use a cell line that's primarily epithelial. Maybe you're going to be looking for enrichment of different downstream genes, like instead of looking at SMAD2/3 like we did here, maybe we'll look at different downstream genes. So yeah, that's I don't think it would be too difficult to take this model and apply it to different cancers. In terms of applying it to maybe like a different phenomena entirely, like stem cell plasticity or differentiation, I think that would be pretty interesting, because overall the overall process is kind of the same. When we're thinking about kind of like iPSCs and differentiation, we're looking at the downstream genes that are influenced by the Yamanaka factors. And so a lot of the work in terms of looking at here we looked at like SMAD2/3 binding, so then it would kind of be a similar process there, where we're looking at the downstream targets of like SOX2, OCT4, all the Yamanaka factors, to see kind of how that reprogramming is taking place. So yeah.
All right, thank you for that presentation. And really quick, before we start the next one, we're going to take a quick group picture. So if everyone wants to turn their camera on, and we can take a screenshot really quick. Okay, smile. All right, thanks for that. Now our next presentation is going to be from Sandy from UT Austin. So if you could start your screen share and begin when you're ready. Judges, I think we're going to have to make these questions a bit shorter. We're way behind, but that's all right. So yeah.
Hi, so my research was actually done under the same research educator as Annabelle and Mackenzie. So you'll be able to see a lot of similar processes. Okay. So my research is about discovering new inhibitors for the papain-like protease of SARS coronavirus 2 using high throughput screening. A little bit of introduction. SARS, severe acute respiratory syndrome, is a respiratory illness that causes COVID-19. So the symptoms range from mild to severe illnesses, including fever, cough, shortness of breath, loss of taste, loss of smell, so on and so forth. Currently, there are vaccines available. As you all know, the Moderna, the Pfizer, and Johnson are one of the main ones. However, due to the new strains and unknown long-term effects, more drugs are needed to be tested. The coronaviral protease, papain-like protease, the one I'm researching, is a essential drug target for viral replication. The reason that I chose that protease is because it's essential. It antagonizes cellular ubiquitination. So that means it's just adding ubiquitin chains to lysine residues, and that helps. It's essential in degrading the defective proteins, which maintains homeostasis. So we don't want to maintain homeostasis. Therefore, targeting those ubiquitin chains might be good for inhibiting the protein. So the main objective is using GOLD, like Mackenzie and Annabelle said, a virtual screening program to identify potential inhibitors for the papain-like protease. So first aim is to find a crystal structure. I did manage to find one crystal structure on the Protein Data Bank website, and it is displayed here on this slide. Second aim is to run Molprobity. Molprobity is a structure validation web service that gives an overview of proteins and nucleic acids. So as you can see on the table, there are a lot of colors displayed. So the ones colored green are the ones that are within the optimum score for that specific criteria. However, the ones that are red they fail to reach the optimum level. So in this analysis, you can see that there are more red colored parts than green. So it might be that the protein itself is not a good inhibitor, but we will still need to test it out with the ligands docking and then see what results we get and then go from there. So our third aim is to obtain the ligands for docking. So one of the things, to obtain potential ligands, we want it to be orally active. And what that means is that it has to be absorbed through the intestinal lining. It has to be carried through the blood, pass through the membrane, and still be able to inhibit the enzyme. And that is done through Lipinski's Rule 5. So that basically says no more than 5 hydrogen bond donor, no more than 10 hydrogen bond acceptor. The molecular weight should be less than 500 grams per mole, and then the partition coefficient should be less than 5. And that's how we got the positive controls on the left side. So there are 5 positive controls. And so we also want to test to see if GOLD is a good docking algorithm. So we also brought in negative controls. So negative controls are the ones that we know won't work or no expectation for them to work on that specific enzyme. So an example could be aspirin. And then on the middle, you can see a cyan colored image of, it's an original ligand of the structure that was obtained from the Protein Data Bank website. So in total, there are 11 ligands that are going to be docked for GOLD. Okay, so aim 3 is to dock into GOLD. I've included a before picture of the structure, the papain-like protease structure. And you can see there's a red box around the particular part. That's where the active site is. And that's where, that's where the ligands are supposed to be docked in that part. So on the right side is when all the ligands are docked into GOLD. So what GOLD does is that it will dock each ligand several times with different conformations and orientations to give the best conformation, so the best binding for that active site. And that gives you a rank, the best fitness rank, so how well it fits into the active site of that structure through the interactions like hydrophobic interactions, the polar contacts, and hydrogen bond also. So you can see I added the top 3 ligands of the top 3 conformations of each ligand. So in total, there are 33 ligands docked into this active site. So now I've included the top 5 best scores in gold. The optimum score for gold is 100. However, you can see from the top 5 that the range are within 50s and 60s. That could be due to the structure itself. That could be due to the papain-like protease structure that we obtained from the protein data bank. Maybe it's an old structure. And also on the table, you can see the ones colored green are the positive controls. The ones in red is the negative, and the original ligand that was in the structure itself is the original. And that's the yellow one. So you can see that the negative control is included within the top 5 of the ranked, and that could also be due to the structure being poor or the inhibitors. Maybe not, the inhibitors aren't good for the structure either. And so on the right side, I've included a picture of the top 5 ligands docked into the active site. So the one colored green is the top one, and then orange, magenta, yellow, and then red. So future directions, we hope to obtain the target protein and the enzyme assay reagents. We also want to express, purify, and characterize the protein so that we want to see how they compare in vitro versus in silico. So acknowledgments. Thank you. That's about it.
All right, thank you for that presentation. If the judges have any questions, you can ask them now.
I just have one question about your downstream assays. So this inhibitor, how are you going to test that to ensure there's no toxic side effects besides what you want to have happen, which is reduction of the replication to the normal cells themselves?
That's a good question. I haven't thought about the wet lab part far ahead because of the whole COVID time, but we hope that there's a protocol for obtaining the assay and expressing the protein. But yeah, that's a good question. Sorry, I couldn't be of more help.
Is your target protein the papain-like protein in the whole coronavirus family or specific to SARS-CoV-2? Is this something that could maybe have broader application to other coronavirus outbreaks?
Yes, it is one of the proteases that helps, has a broader application to other coronavirus. There's also the main protease. There are a lot of other proteases, but they all have a broader application.
What got you interested besides, like, if it was just like COVID happening, that makes sense, but what drew you to doing this, and what do you plan to do with the experiences of working with this type of research moving forward?
The reason that got me to do this is that I've read in the past readings that viruses have become more prominent with the recent years. Tuberculosis outbreaks, others that don't come to mind right now, and COVID, they have been more prominent in the recent years. And so I think it is really important to get into research like this because a lot of stuff will begin to happen. A lot of strains, a lot of viruses will begin to have an outbreak, and we have to control it as soon as possible. In terms of for future research, I do want to continue with screening and viruses. I think it's a learning opportunity for me to about in future for med school and PhD, hopefully.
Thank you.
All right, thank you so much. We have one final presentation from Arvin again about some other research he's doing. So you can screen share again and begin when you're ready.
Sounds good. All All right, hey guys. So I'll be presenting another talk titled "The High Throughput Immunoassay Identifies a Panel of Candidate Serum Biomarkers for High-Risk Prostate Cancer." And so this is some work I did with the Canary Center for Cancer Early Detection at Stanford. So prostate cancer currently is the most common cancer in men in the United States. It accounts for 19% of all new male cases of cancer. And has a 29% 5-year survival rate if metastasized at the time of diagnosis. However, it only accounts for about 8% of male cancer deaths. So, a lot of this, if we're looking at this 8% of male cancer deaths, this metric, it's a relatively low mortality rate. And a big reason for that is because of the current gold standard of detection, which is prostate serum antigen. And so PSA is basically a protein that's secreted, that's very, very highly enriched in prostate cancer. And so it's very easy to do just a serum ELISA test for PSA and basically be able to identify cases of prostate cancer in men. However, if we're looking at this infographic here, we can see that it's basically a study comparing 1,000 men who were not screened using PSA, and then 1,000 men who were screened with PSA. And these dots here with P's in them basically represent the men currently dying from prostate cancer. Same thing here. However, when you screen with PSA, we can see that all of these blue dots are people that didn't have cancer but got a false positive. And then these dots with X's in them are people that didn't have cancer, but still got diagnosed and treated for it unnecessarily. And so this is a huge problem because if we're thinking about, what the implications of this are, is that a lot of the times just getting a biopsy can lead to bleeding, internal bleeding, and just overall discomfort for the patient. And now thinking about these men that were diagnosed and treated unnecessarily, obviously chemotherapy and cancer treatment can be extremely taxing and be a detriment to overall health. And so we really need a detection technique that not only detects prostate cancer, but can do so in a way that stratifies indolent from extremely aggressive prostate cancer. And so in order to identify biomarkers for this cause, we recruited patients from the Stanford University School of Medicine, from the Stanford Hospital. And basically we collected serum from these patients while they had prostate cancer before radical prostatectomy, which is removal of the entire prostate, which is pretty commonly done in prostate cancer patients. And then we also collected serum right after the surgery. And so we ran the serum through a mass spectrometer. And here we analyzed all the proteins shown on this heat map here in the blue are going to be proteins that were enriched in the preoperative serum sample and then went down in the postoperative serum sample. And so we picked out this panel of biomarkers for further validation and analysis. And so looking here at the expression of these various proteins in prostate cancer clinical data sets, we can see that a lot of these proteins are upregulated in prostate cancer, specifically A2M, AMBP, LCP1, and PPBP. Apart from just looking at mRNA expression, we wanted to basically take it a step further and look at whether we can get any sort of kind of spatial data from this. And so we utilized a data set in which the researchers basically took a whole prostate and a pathologist graded the different tissue sections. And so here we're seeing some tissue that's marked as cancer Gleason 3+3. And so Gleason scoring is basically just a measure of how aggressive the cancer is. And so 3+3 is going to be isn't going to qualify as high risk. Rather, 3+4 is. So any Gleason score higher than 7 is going to be considered high risk. And so basically these researchers took the prostate, took these sections and placed them on these microarrays. Then they were able to construct these kind of spatial transcriptomic maps to see where expression of various proteins are enriched. And so we went ahead and took this data, processed it, and queried our proteins of interest in this data set. And so looking here at A2M first, when we're looking at normal prostate, we can see overall there's lower expression as indicated by the blue. Then looking at inflammation, because inflammation can mirror a lot of the kind of similar morphological changes that's seen in normal and high-risk prostate cancer. We see that expression remains low. However, in the Gleason 3+3 and 3+4 prostate cancers, we see that expression increases markedly. Looking now at LCP1, we see that expression kind of remains pretty low throughout, whether it's normal prostate inflammation or any sort of aggressive prostate cancer. But looking at CST3 here, we see that it's just kind of everywhere at every stage of, at every different type of tissue. So in the normal prostate, it's fairly highly enriched. Inflammation, it's still there. And then Gleason 3+3, you can kind of see it. It just looks like it's kind of on fire. There's a whole lot of expression there. Also, that's also the case in Gleason 3+4 as well. AMBP, very, very low expression. And ApoA2 was only detected in one Gleason 3+4 tissue section at all. And so moving from the spatial transcriptomics, we basically wanted to look at whether there's any particular clusters of cells that are responsible for secreting these proteins. And so we analyzed a single cell RNA-seq data set that was done on various high-risk prostate cancer patients. And so we, using T-distributed stochastic neighbor embedding principal component analysis, we were able to determine basically these clusters of cells that are transcriptomically very similar. And looking at the expression of our various proteins of interest in this data set, we can see that A2M is enriched in these two clusters, cluster 4 and cluster 8. And so these are clusters of epithelial cells. LCP1, we can see there's very little specificity to how it's expressed in these various clusters. CST3, we see that, again, there's high expression kind of everywhere. And this, again, reflects what we see in spatial transcriptomics data, where we see that there's high expression regardless of the Gleason stage and also the prostate cancer status. And then AMBP, again, reflecting kind of what we're seeing in the spatial transcriptomics data, there's very little expression anywhere. And so looking at now endothelial cells that are present in the tumor. So basically the support cells and a lot of the vasculature that's going to be present in the tumor, we wanted to see if there's robust expression of any of these biomarkers in those cells. And we can see here that A2M is fairly enriched in these endothelial cells, whereas in the epithelial cells, it's relatively low. There's only kind of 2 clusters that are mainly expressing it. LCP1, again, kind of mirroring the spatial transcriptomics and epithelial cell single cell RNA-seq data. There's very little expression anywhere. CST3, again, follows that same kind of trend. There is clusters 0 and 1 that are kind of more highly enriched for CST3. And AMBP, we're seeing, again, very, very little expression here. And so from this endothelial cell population, we decided that we wanted to look at whether, there was add another layer of another dimension to this analysis and basically see if there's any particular time point in which expression of these these proteins increases. And so using a technique known as pseudotime differential expression analysis, we were able to analyze this. And so basically what we're doing in this case is we're taking the endothelial cells, and all the endothelial cells are kind of going to be in distinct populations. But on top of that, they're also going to, within those populations, they're going to be at different time points of transcriptomic progression. And so we can kind of track the progression of a cell from just a normal endothelial cell into what's called an activated endothelial cell. And so an activated endothelial cell is going to be cells that actively contribute to the growth of the tumor and facilitate cancer aggressiveness. And so starting here at this cluster, clusters 0 and 1, those are going to be the normal endothelial cells. And then moving outward into these branches into clusters 2, 3, 4, and 5 are going to be the activated endothelial cells. And so we can see here that A2M, when we're looking at this, we're starting with the lower expression in the normal endothelial cells. Then when you have the activated endothelial cells, the expression goes up. LCP1, there's little expression. CST3, we're kind of seeing the opposite pattern as A2M, where the normal endothelial cells have higher expression, and then as they become activated, they have lower expression, and then AMBP, again, reflecting the pattern that we saw in the past with very little expression. So now looking at some future work, we'd like to do some further validation of these serum biomarkers showing the most promise. So that's doing more bioinformatic analysis, but we'd also like to do a pairwise comparison of a large patient cohort of these serum samples to better validate their diagnostic ability. We'd then also like to do immunohistochemistry of the patient tumor samples. So basically seeing if there's any protein localization of these biomarkers in the tissue sections. And then we'd also like to probe the mechanism behind A2M and CST3 modulation in these activated endothelial cells. And so with that, I'd like to thank our collaborators, James D. Brooks in the Urology Department, Sharon Pateri at the Canary Center, and the Stoyanova Lab, and the funding sources without whom this work would not be possible. And so with that, I'd like to open the floor for questions.
All right, thank you.
Yeah, so I just had a question really quick. It's more of a overall question, not specifically towards these data, but I was just curious. It seems like you are doing quite a bit of bioinformatics, and I was just curious, what bioinformatics skills or coding skills did you have coming in versus what skills did you learn whilst you're doing the research?
Sure. Yeah, that's kind of funny because, you know, I kind of came into all of this not knowing any bioinformatics. I kind of had to transition all my projects to being primarily computational because of COVID, because, you know, I wasn't able to go into these labs a lot of the time. And so these are all skills that I kind of had to just pick up. I had a little bit of background in Python, but learning how to handle these data sets, I had to pick up R, and then also learning how to use a lot of these tools, like in my previous work with Max Sped tools, and then also with this single cell RNA-seq data that's kind of, it has its own kind of host of bioinformatic tools that are necessary. So I had to pick those up as well. So it was a steep learning curve to transition from being like a bench researcher into being, you know, primarily bioinformatic, but it's been a rewarding journey, I think. So yeah.
So I know you talked about some of your, you know, like what you would do next steps, but a little bit more big picture. If you're, you know, going to, I don't know, like NCI or someone to say, hey, let's change the recommendation from PSA as a biomarker to one of these biomarkers, what would be like an ideal study that you'd want to perform? What would be some criteria that you would want these biomarkers to meet before that transition was made?
Right. Yeah, that's an excellent question. So I think the burden of proof to replace PSA is a high one. It's PSA is highly touted as, you know, kind of one of the, everyone's trying to find the PSA for their kind of own cancer, you know, and so being able to replace PSA is a tough task, but I think it would come more likely in the form of kind of a panel rather than a particular kind of silver bullet candidate. I think it would involve more kind of a panel of 2 or 3 different biomarkers that hopefully would mirror the sensitivity and specificity of PSA, however, reduce the number of misdiagnosed cases and also be able to cluster indolent from aggressive disease. And so I think validating this in a larger cohort is definitely necessary. Looking at not just serum expression, serum levels, but also proving that it's present in the actual prostate cancer luminal cells is very important. And of course, you need the actual, if you want to bring this into the clinic, you need the actual ELISA and the actual diagnostic tests that have enough sensitivity and have been validated to be able to bring this into the clinic. So, yeah.
All right, if no one has any other questions, then that concludes our research division. Thank you so much to all the presenters. You did a great job. We went a little bit over, but currently happening is the grad school fair. So if y'all want to hop on over to that, the Zoom link should be in the program. Feel free, and then I will stay on the Zoom call for the judges if you want to deliberate a bit. All right, so feel free to, if you guys want to talk about it or just like have time to write out your feedback. I know it's hard to do while listening at the same time.
Superpower of ADHD is I do both.
Oh, I cannot. I have to focus and listen. I can't do both. I don't multitask.
I do 5 different
in real time. I don't.
Yeah.
I can't do that in real time. I have to focus. But I was able to, during the questions, respond to the questions that were asked, not in depth as I would have liked to have provided feedback since it was real time. But I think overall, I'm a bit biased, just to be honest, because